Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is titled Biostatistical Challenges of Trials in Rare Diseases. My name is Nima and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for future download. At this point, I'd like to thank IDDI, who helped to develop the content for this presentation. The International Drug Development Institute, IDDI, is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Raleigh, and San Francisco. Now I'd like to introduce our speakers for today's event. Professor Hilgers graduated in mathematics from the RWTH Aachen University and received his PhD in statistics at the University of Dortmund while working at the time at the University of Cologne. He has been professor in biostatistics and chair of the Department of Medical Statistics at the RWTH Aachen University since 2001. Professor Hilgers gives biostatistical advice to clinical and experimental trials in all clinical and preclinical areas. From 2013 to 2012, he coordinated the IDEAL project funded by the European Committee, which established new methodologies for small population group trials. Everardo Saad has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo, and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology, and he has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. So now, without further ado, I'd like to hand the presentation over to our first speaker. Everardo, you may begin the presentation when you're ready.
Thank you very much, Nima. On behalf of IDDI, I would like to welcome you all and thank you for attending this presentation. So today we have the privilege of having Dieter Hilgers with us. As Nima presented, he has developed tremendous experience in trials among small populations and rare diseases and orphan indications. We'll see a little bit about the meaning of these terms pretty soon. So he will conduct most of the presentation, in fact, and my task is really to provide a short introduction in which I will present some relevant definitions, some facts and figures that I could gather, and some historical background in this field. Whereas he will then come over and discuss some topics, some of which shown here, the public research interest in R&D, the state of the art in this field. He will illustrate a lot of his presentation with the examples of the IDEAL project, which he will describe to us, and at the end he will provide some tentative conclusions. So just some facts and figures about rare diseases in general. Of course, each of these diseases is rare, but collectively they are not. So nowadays people talk about anything between 6,000 and 8,000 different conditions being recognized as rare diseases. such that in Europe alone, around 30 million people are affected. In the US, anything between 25 and 30 million people. So common estimates are of around 6% of the European population, up to 10% of the US population, depending on the source we look at. So since many of these disorders have a presumed genetic basis, many of them affect children. And this is a very unfortunate fact that nearly 50% of these rare diseases affect children. And many of them don't really live to see their fifth year. So many of these disorders are potentially fatal, although some are not, and some are exclusively adult disorders, but many unfortunately affect the pediatric population. So, of course, the central problem here is the rarity of individual diseases. And just to give you a flavor of what people mean by rare, depending on where you look, if it's in the US or Europe, anything that affects in Europe less than 1 in 2,000 people, or 5 in 10,000, or 50 in 100,000 people is considered rare. And just to give you an idea, so keep that number in mind, less than 1 in 2,000. I will tell you the numbers in the US later on. So since all of these are rare disorders, of course, we expect a few cases, and this creates several problems for drug development, for clinical research in general. So these diseases have a diversity of settings in which they can present. They can be very heterogeneous in their presentation, depending on mode of penetrance and many things related to genetics and to the environment. There is often difficulty in diagnosing, in really knowing who the patients are. There is, of course, relatively poor knowledge about the natural history of these diseases, just because they are rare, and many of them have not been investigated. And in fact, many continue to be described recently. There is little experience with endpoints and very little characterization of these endpoints for clinical trials. So for some conditions, people don't know exactly which endpoints are really determinant, which are really indicative of the disease process. Of course, the diseases being rare, there is a lack of standard treatments, and therefore lack of suitable controls for controlled studies. Patients are dispersed geographically, and this poses tremendous difficulties in terms of running clinical trials. So the resources in general are dispersed, the most important of which the patients themselves. And last but not least, for developers, for sponsors, for drug companies, there is the small expected return given the size of the market. And in fact, this has been underlying all of the legislation related to rare diseases. That in fact provided the impetus for creating specific legislation in order to encourage potential developers to invest in these conditions. So really the first legislation that is known came in 1983 in the US. So this was the so-called Orphan Drug Act, and this was created really to provide incentives for drug companies mostly to develop drugs for rare disorders. So there was a need to create a definition of orphan conditions. Then in some countries later on, for example, Japan 1993 and Australia 1998, special legislation was created as well. And in the European Union, the action really started mostly in the year 2000, when the EMA created its Committee of Orphan Medicinal Products, or COMP. And this is the entity that grants orphan drug designation to drugs that may one day be potentially commercialized as a drug for a rare, or a treatment for a rare disease. And by the way, all the legislation speaks about not only treatment but also prevention and diagnosis. So they are all the three aspects are contemplated in these legislations. And then after this, the entities created their own sets of guidelines for drug development or guidance for industry, for example, in 2006 in the EU, and more recently 2015 by the FDA in the US, a document about common issues in drug development in rare diseases. So just to put very simply the US perspective and the EU perspective in 2 slides, and then I'll hand it over to Dieter. So the U.S. perspective is such that the legislation, like I said, was created some years ago, and at the time the threshold for defining a rare disease was the number of people affected in the U.S., and this number is and continues to be 200,000. So taking the population of the U.S. in 1983, this is 1 per, let's say, 1,200 people, and with the current U.S. population, this is more like 1 in 1,600 people. So that's the definition of a rare disease in the U.S. Now, interesting, the orphan drug designation is not only granted to rare or orphan diseases, but also to situations for which the costs of developing and marketing are not expected to be recovered. But this, of course, has to be analyzed by FDA officials. So it's there is a provision for orphan drug designation to be given to drugs that may target a larger population, as long as there is no expectation of receiving of recovering the investment. Now, this has been apparently a very, very successful legislation in the US, and the program that started with the Orphan Drug Act has enabled the development and marketing of more than 600 drugs and biologics since 1983, compared with less than 10 such products in the decade before the legislation was put in place. Now, in Europe, like I said, COMP is part of the EMA, the Committee for Orphan Medicinal Products. It's the committee responsible for recommending the orphan drug designation. And in Europe, rare diseases are those that affect less than 1 per 2,000 people or 5 per 10,000. And a lot of action is ongoing in Europe as well. Since the year 2000, more than 1,100 orphan drug designations have been granted, although most of them did not turn into a marketing authorization because the development is lengthy and many things can happen along the time. But the latest figures that I could get are from about 2 years ago, when 114 orphan drugs have already received market authorization in Europe. So this is my introduction, and I'm pleased to hand it over to Dieter, who will say more about rare diseases.
Okay. Hello, everybody. My name is Dieter Hilgers, and I was already introduced. Can everybody hear me? Okay. So, yes, I will go to transparency number 7, where my talk is starting. So I will briefly say something about the public research interest in rare diseases from the European perspective on. So following the EU Cross-Border Healthcare Directive from 2011, rare disease seems to be an excellent showcase to that. And as Everardo already pointed out, the deficits are obvious in rare disease care and research, which consist in the majority of rare disease lack of accurate diagnosis and efficient treatment on one hand, and it takes a very long time for new therapies to become available for patients with rare diseases. Nowadays, all the people in the European community think that developing solution requires sharing data and collaboration internationally, as well as globally, from the patient's point of view, as well as from other initiatives' point of view. And I come back to this in a couple of minutes. Our overall aim is to improving the life of rare disease patients, and that requires cooperation between research and healthcare organizations. So as already pointed out by Everardo, you see the numbers here, and I have something to say, because rare disease definition seems to be very obvious. For example, when you think about Duchenne muscular dystrophy, or Huntington disease, or Friedreich ataxia, or acute myeloid leukemia. But there is a great debate arising concerning the Salemi slicing, which means that you have a biomarker defined subgroup of a common disease and define this as a rare disease. And so you open up the door for drug legislation if you relax the condition. So I think that is a very critical point for the regulators in that context. Well, what did the European Commission does in the past? Well, from 2007 to 2015, in the FP7 and Horizon 2020 program, they spent about 900 million euros for funding research in that direction. And that belongs to around 164 projects. And the 164 projects are gathering a critical mass of rare diseases, disease cases, and expertise from collaboration. On the one hand, the prevention of treatment and diagnosis of rare diseases. On the other hand, the third axis is tools and challenges relating to effective and ethical medical data sharing. And the fourth hand is new approaches for clinical trial in small populations. And I will come to this in a couple of minutes, but let's go ahead with talking about the tasks which you have to address for rare disease research. So what is pointed out in a brief paper, in a current paper by the European Commission, the unmet medical need. So you have 5 tasks to address, and that means improving diagnostic, prevention, and treatment of rare disease, facilitating the regulatory pathway for potential treatments, effective and equal provision of health care for patients with rare diseases, effective management and pooling of research and medical data, and contributing to and benefits to global collaboration. So you see that this also has some relation to the European market or political aims the European Commission has in mind. So we'll come up to the point facilitating the regulatory pathway. And if you think about this, the explanation in the paper, which is delivered by the European Commission, starts with the major challenge, and that is to develop novel approaches for clinical trial methodology for small patient population to facilitate demonstration of safety and efficacy of orphan drug candidates in order to attain marketing authorization and enter clinical practice. And this task is a continuation of the research, which is funded by the 4th aspects on my transparency number 11, and which leads to funding of 3 collaborative projects in the year 2013 to 2016. But before I come to these collaborative projects, and in particular to my ideal project, I will give you the global initiative which is behind research. So we are not only thinking in European context, we are thinking globally since 2009, where the European Commission, together with the National Institute, founded the collaboration which is called the International Rare Disease Research Consortium, IRDIRC in my spelling, which has an initial kickoff meeting in 2010 in Reykjavik. And well, the IRDIRC is a very active group, which has first formulated an initial vision for the decade 2010 to 2020, and they would like to develop 200 new therapies in that decade. And we can say, unfortunately, or surprisingly, this was already reached in 2016. So they had to update their goals, and this was done in 2016, and they had 3 new goals. And the first goal addresses the diagnostic process, which should be independent from countries you are living in. So this reflects also the cross-border healthcare initiative. The third goal also addresses methodologies which have impact on diagnosis and therapies. And I'm very interested in the second goal, which states, okay, you have to develop 1,000 new therapies for rare diseases in the next decade, starting from 2017 up to the year 2027. Well, of course, you might say, okay, this cannot be done by funding of particular projects alone. So now we have to collaborate, and this, I think, was when the European Commission launched a call, which is finalized in April 18 this month, which is called the European Joint Program on Rare Diseases. And they would like to address the overall objective to combine all these initiatives and projects and this research and knowledge, which was funded up to now by European projects, to one initiative, including, for example, the RRNs, which is the abbreviation for European Reference Networks, which combines around 900 hospitals in the European Commission, and the IRDIRC, with the patient advocacy groups, and so on. And the project will be funded by 55 million euros for over 5 years. And I'm looking very much forward if the funding is going on, and if our project will be funded. It is coordinated by the INSERM, and I'm involved in that project. I'm looking very much forward for it. So now we're going to talk about the state of the art and in the direction of trial methodology. And well, as Everardo already pointed out, in 2007, the EMA guideline was released, formally called from the Committee of Medical Products for Human Use, CHMP, and the European Medicines Agency in that time is called EMEA. So, but now the guideline was adopted. And it is very interesting to note that it is pointed out on the first page that no method exists that are relevant to small studies that are not also applicable to large studies. So this is a gap of knowledge. So we do not have a unique method for small population group trials. However, it may be that in conditions with small and very small populations, less conventional and less commonly seen methodological approaches may be acceptable if they help to improve the interpretability of the study results. So this is the starting point. I think that was in mind when the European Commission released or launched the call on new methodologies for clinical trials in small population groups, which should develop new and improved statistical design methodologies for clinical trials aiming at an efficient assessment of safety and efficacy of a treatment of small population group, in particular in rare diseases and personalized or stratified or individualized medicine. So independent of the specific kind of disease. So that could be oncology, that could be on neurology, nothing is stated about that. So the framework of the collaborative project should be multidisciplinary, relevant stakeholders, patient advocacy group, industry, and regulators should be on board. Ideally, the results should go into a clinical trial guideline, and collaboration with relevant organizations outside Europe is welcomed. And the expected impact, as you can see, is cost-efficient clinical trial. And at the end, there were 3 projects funded in that 7th framework initiatives. And I start with the upper one. It's the IRDIRC project, Advances in Clinical Trials Designs and Clinical Innovative Excellence, coordinated by Kit Roos from University Medical Center of Utrecht. And that project aims to develop design and analysis methods for single trials and series of single trials, in particular where patient-level information and perspectives are going into design and the decision-making, so that that they result in improvement in regulatory purposes. And at the bottom, you find the INSPIRE project coordinated by Nigel Stellert from Warwick University. The INSPIRE project is innovative methodology for small population research, and it addresses 4 aspects: early phase dose finding studies, decision theoretic methods, and confirmatory trials in small population clinical trials, as well as use of evidence synthesis in planning and interpretation of clinical trials in small population and rare diseases. And in the middle, you find the project which is coordinated from myself, the integrated design and analysis of small population group trials, abbreviated as IRDIRC. And we are focusing on several aspects like randomization, extrapolation, adaptive designs, optimal design, and so on. I will come to that in a couple of minutes. And try to integrate all of these points to improve the design and analysis aspect as far as we can do that in 3 years of a funding period. So before I come into detail to this point of view, I would like to briefly go back to the IRDIRC initiative. The IRDIRC has launched 8 task forces which created to tackle specific topics within rare disease research. And as you can see, in the year 2015, middle of 2015, a small population clinical trial task force was created. I'm very happy to join this task force. The objective is very similar to the objective what is written down in the FP7 call. And as you can see in the steering committee, not only Kit, Nigel, and me were involved. There was also Simon Day and Irony from the FDA and Christina Larsen from the AMA. So a very strong group. And we had a very fantastic meeting in 2016 in London. And the report can be downloaded from the IRDIRC website. And briefly saying, there are a lot of design aspects which are written down in the report, which may be considered in the context of small population clinical trials. So, for example, adaptive randomization, Bayesian design, crossover design, factorial design, enhanced trial design, group sequential design, and so on, whatever you see there. So a lot of very specific topics. So Kit, Nigel, and me have written a paper in the Orphanet Journal where we formulated what we thought in our projects in a joint manner, what are the directions of new development in that context. And we broke that down in the first part in the areas level of evidence, pharmacological consideration, choice of endpoints, and methodological and statistical considerations. That is very parallel to the EMA CHMP small population clinical trial guideline, which I mentioned just before. But more to that, we added the point extrapolation, because that was an upcoming discussion which addresses the point of the reflection paper on extrapolation of the EMA, where some rules were defined for pediatric clinical trials. And we also added a point on patient involvement and ethical consideration. So another paper where from the IDEAL project, we stated some of our first recommendation was a paper concerning design and analysis in clinical trials in small rare disease populations in the Rare Disease Journal. And well, I will only well, we give some recommendation about some very specific points, but I will focus more on what we call the three-level approach. We think that on the first hand, there should be a rigorous application of already developed efficient design and analysis techniques, like, for example, using ANCOVA models instead of evaluating the difference between the last endpoint, the last observation to the former endpoints, or using longitudinal data if you have that in mind. So these are rigorous applications. Evaluation. The second level is evaluation of the traditional level and show where the methods fail, because if the sample size is going down. And the third level is new methodologies which are never thought about for clinical trials in small populations. And we address in the IDEAL project all these three areas. And but before I'm coming to details from the IDEAL project, I would just briefly mention one of the papers and which one of the approaches which you see very frequently. So there is a paper, for example, that of Cornu. There's also a paper of Gagne and Gupta and Perma. Well, all these papers look to the literature and report about which design is already used. And most of them are coming to parallel group design, crossover design, factorial design, and one trials, then randomized placebo-phase trial, randomized withdrawal trial, and some specific like stepped wedge design or something like that. So this is already new, known design, but there is no innovative design in this paper, which you may find in the publications we have. So what is IDEAL in more specific ways? So IDEAL is broken down in 9 work packages. It deals with assessment of randomization procedure. The work packages coordinated by me. So then Holger Dette in Bochum do research in extrapolation of dose response information. Then the design of pharmacogenetic trials is done by Stephen Sen in the Luxembourg Institute of Health, and adaptive design studies is investigated by Franz Koenig from the Medical Center in Vienna. On the left-hand side, you see simulation of clinical trials, which is done in the pharmacometrics group by Mats Carlsen, Uppsala University. Malgorzata Bogdan from Warsaw University is dealing with selection of genetic factors. Then this very active group of around Gert Molenbergs from Hasselt and Leuven University are looking to surrogate endpoints in this group. And if you go into the right side, you see, well, optimal design for mixed models are investigated by Francois Montré in Paris, and Carl Frederick Bormann from Chandler's University. He's also affiliated to AstraZeneca, has investigated decision analysis. And briefly, the summary is, well, we published in this 3-year period around 70 publications in peer-reviewed journal. That is the current state. We are not at the end. And this is in various perspectives. There are applications in Lancet Neurology, for example, and there are papers in the Annals of Statistics as well. So we had a lot of presentation, 170 around, so I must count 171 now, because this is another presentation I'm giving now. We have a series of 11 webinars, which is available at the website. We have 15 released various free available software programs, which can be attached also via our IDEA website. And we have already given input to clinical, to rare disease clinical trials, and formulated 33 recommendations to conduct small population clinical trials. And the website is given below, www.ideal.rwth-aachen.de. And there you can find all the outputs, and most of the publications are open access, so you can act them. So briefly, because otherwise I'm running out of time, I can tell you a lot of things about the IDEAL project. The randomization, concerning randomization, we developed a completely new view on randomization and selection of the best practice randomization procedure. This selection process is taking possible bias into account. So we have 3 recommendations in that direction. The recommendation you find in another paper, which is under review in the Orphanet Journal, unfortunately for 3 months, because the editorial stuff is changing. And the numbers behind these recommendation refers to a literature list, which I would like to submit to you with this presentation, so that you can see which of the recommendation belongs to which paper, where you can find more details in. So Holger Detzel develops in the extrapolation context new optimized design and analysis strategy for comparing dose response profiles to extrapolate clinical trial results from large to small populations. So extending the knowledge of a larger population to a smaller population. That is basically what he do. And Franz Koenig addresses statistical methods to adapt the significance level and allow confirmatory decision-making in clinical trials with vulnerable small population clinical trials. So some aspects concerning extrapolation, some extra aspects concerning what he called, if you have knowledge in single-arm trial, is that enough to already confirm that the drug is working? So Francois Montré is working on optimal design. For evaluation, so she developed design evaluation methods enabling small clinical trials to be analyzed through modeling and continuous or discrete longitudinal outcomes. And Stevenson, who is doing the pharmacogenetics part, developed approaches for planning and analyzing trials for identifying individual response and examining treatment effect in small populations. Work package 7, which is called the simulation part from Mats Carlson. He developed new methods for sample size calculation and type 1 error control for model averaging and parameter precision in small population group trials, if you are using nonlinear mixed effects modeling. Work package 8, Margosata and Bogdan, is dealing with genetic factors, and she developed new methods for identifying biomarkers or prognostic scores based on high-dimensional genetic data and small population group trials. And work package 9 is the decision theoretic part, maybe very interesting for you, for those of you who are working in the drug companies, because Carl Frederick evaluated how to optimize the overall value of a drug development program to patients and to regulators and societies under opacity and regulator and payer rules as well as very rare diseases. So last but not least is the biomarker aspect, which is investigated by the group of Gert Molenbergs in University of Hasselt and Leuven. He developed methodological aspects to evaluate potential surrogate markers and to analyze data from small numbers of clinical trials and to emphasize on fast and easy computational strategies. So if you would like to have an overview of our recommendation, I would like to draw your attention to the picture. We arranged this in more the development phase, and so you can see on the left-hand side, as an overall umbrella, the decision theoretic part, and on the left-hand side, the information you have at the beginning of the trial, so generating prior knowledge from existing data, and put that in the design and analysis phase of single-arm trials, comparative trials, where you have longitudinal data, dose response information within subject design and level of significance. And to see how this relates to the IRDIRC task force, I would like to draw the attention to that part. We identified 73 items from the IRDIRC report, from the IRDIRC task force report, and 61 of these are already mentioned in publications which are written down by the IRDIRC group. And if you relate these topics, which are written in blue there, to the work packages, that is depicted here. So you can see that all these aspects are nearly addressed by at least 3 or 5 of the work package. So that makes the topics as an integrative point of view. So what do you have to know if you would like to design an ideal small population clinical trial? So what is ideal capacity? And that is depicted here. You see the work packages in the middle, and you see in blue some of these skills, biostatistical skills you have to have in mind. So on the top, adaptive combination test, or real world data, or pediatric investigation plan, or for example, false discovery rates. All these aspects are mentioned by our project. And on the far right, you see in capital letters the ERDO, which means the evaluation of randomization procedures for design optimization. So already you have to be familiar with some new aspects as well. So for the sake of time, I cannot go into more detail. So let me end with some perspectives. So what do I think what is necessary now? What we have to have on board? Well, we need to have well-trained biostatisticians, and we have to extend our methodological methods. I agree with the EU funding in that direction. And we have to evaluate much more complex design, which are now in mind of most of the people, like platform trial, basket trial. And these first 3 projects, so let's keep fingers crossed, are part of a grant application we submitted to the European Commission together, where already the IDDI was involved in this called the PERSIST project. Then we are thinking about grouping rare diseases to get a richer population where we can investigate rare diseases. We are thinking about evaluation of patient-centered outcomes and natural history study, and this was in the focus of the European Joint Program, which is already submitted by the INSERM. Daria submitted that, and well, let's keep fingers crossed that this is already also funded by the European Commission. And also one aspect which I think which needs major rethinking is that we have to think about limited population designs. By that I mean, if your population is worldwide of size 8, and you would do something, you would like to do something for this, you cannot think that population-based analysis, like what is behind Bayesian statistics or something like the usual frequentist method, would be sufficient to think about. Maybe we have to go back to Fisherian statistics in that direction. And with that, I would like to thank you all for listening, and I would like to give back to Nima.
Well, thank you very much for your insightful presentation. Now I would like to invite our audience to continue sending in their questions or comments using the questions window for the Q&A portion of this webinar. I've already received a few questions, so let's start with those. So our first question here is, what are the differences in regulatory requirements for drug approval for rare diseases?
So, Everardo, well, I will go ahead. I will start.
Okay.
I think that if you think in terms of regulatory requirements for rare diseases compared to more frequent diseases, then most is slowed down by the Orphan Drug Act, as well as what is written in the orphan designation in the European context. So mostly that allows pharmaceutical companies to benefit from incentives from European Union to develop a medicine for rare diseases. So reduced fees, good consultation and protection from competition once the medicine is placed onto the market. But mostly these things are assessments which are based on the prevalence. I mean, if you are thinking about Friedreich ataxia, which is supposed to have a prevalence on 1 to 40,000 cases, which translates in the European community to around 12,500 affected patients, then there must be a difference to if you think about a ultra-rare disease, which has a prevalence in 1 to 1 million or 1 to 100,000, which addresses only 1,000 affected patients in the European Commission. And the COMP is doing it on a case-by-case basis. So they are hardly working on putting more transparency in their decision rule, but that is a hard work to do. And I don't think they have strict rules how to judge this. And in that direction, I would like to address your focus to Work Package 9 from Karl Friedrich Burmann, who makes evaluation in that context. Maybe, Everardo, you would like to say something?
Well, no, not really. I'm sort of a newcomer to this topic. The things that were quite clear to me when preparing for this presentation is that there are some misconceptions out there that might lead some people to think that developing drugs for rare diseases might be somehow easier, and that's not the case. I mean, that it's not necessarily faster, it's not necessarily cheaper. It's true that there are some incentives in terms of market exclusivity and different regulations in different places, but the overall notion that I get is that it's not so easy as it might seem. I don't know if you agree with that statement, Dieter.
Yeah, I agree.
Yeah, from the point of view of a sponsor, so it's not because you know, you can deem something to be, you know, to have orphan designation, that it will make your life easier. It might be the opposite. But that of course is not to discourage, because we need you know, as much as possible investment in this area. But it's just that there appears to be some misconceptions as to the eventual ease with which, you know, the approval will be conducted by regulatory agencies.
Yeah, yeah, that's even so my impression. I mean, you must take into account that the drug legislation has a long history for not rare diseases, so more frequent diseases, and that it is a rather young area for the rare diseases. And I think that the regulators are very conservative in their thinking and are very hesitating and open the door to relaxing all the conditions. So they need approval of new methodologies so that for the new statistical methodology, so that you have shown that it makes sense, and that takes some time. You know, that's the point. So that's why we don't have very much input in that. But let me say that the EMA is expected to make a revision of the small population clinical trial guideline, but it takes some time because you already know that we have the Brexit, and for this reason there's some movement of the EMA, and now all are focusing on storing anything together and moving to Amsterdam, for example, than to making a new guideline. So let's wait some time.
All right, thank you so much for those answers. Our next question here is, do your programs include input from payers, since many seem reluctant to accept small or unconventional study results?
Yeah, that is a very good point. But unfortunately, I have to say we don't have the chance to include the payers, neither in the IRDIRC project nor in the INSPIRE project. We have some contacts to health technology assessment people in that context, but that's not directly the payers. Well, when you submit your proposal, you have to fix all the people who are involved in the project, and you cannot say later on, oh, okay, I would like to involve these people as well. And all these people, payers and the regulators, are very hesitating and saying, okay, I be part of a proposal which I never know will be funded. So yeah, that maybe that was the reason why we did not be in contact with payers at that point. But even in that case, Carl Friedrich had included this in his thinking.
Thank you so much for that answer. Our next question here is, how do you deal with patient heterogeneity in these trials? Can you conduct subgroup analysis?
Okay. Well, that refers to another guideline which is already under revision, I think, at the EMA, the subgroup analysis. Well, a very interesting point. Well, most of the people think that stratification in randomized clinical trials is the solution to that, even that is mentioned in the EMA guideline. Well, I'm a little bit hesitating because stratified randomization, which goes before the stratified analysis at the end, does not show so beneficial in our investigation about the randomization processes. So let's wait some time, then we had a paper finished in that direction, but I'm a little bit hesitating. And what I would like to say is that I think there are some very contrary discussion at the moment, because there's recently a meeting at the FDA where they are talking about rare disease clinical trials, and they recommend basket trials. So several disease patients are involved in one group, and that includes more heterogeneity from my perspective on. So I think the point of heterogeneity is not solved at the moment. I think at the end that goes to a discussion about the level of evidence. So accepting more heterogeneous population in your trial, that would mean that the sample size is not enough to show a particular difference, and then we need other evaluation methods to evaluate your analysis. And from my point of view, that is the point where not Bayesian analysis is coming into play. I think that is a point where randomization-based inference is coming into play. But even that, we would like to evaluate that in the PERSIST project, which is under review at the moment, and let's keep fingers crossed, and I can tell you something in the next years.
Thank you for that answer. Our next question here is, how can surrogate endpoints be validated in rare diseases?
Well, I think I have to answer that as well. Well, I must say I'm not the expert in that point, because I was very happy to be in contact with Mark Büse and Fred Molenbergs from the Leuven and Hasselt Group. Very nice guys, very smart guys, where I can pose so many questions that I've learned about so many. But what I understand at the moment is that surrogate endpoints is not only surrogate endpoints. The methodology they developed could also be applied to so-called PCOMs, patient-centered outcome measures, to evaluate. And so Fred will evaluate this in the European joint program if we are successful in that direction. And what I understand is that Fred said to me that the causal inference approach is the best one if the sample size is going down. And Fred and his group found methods for treating numerical instabilities as well. So please have a look to the papers of Fred and his group, and probably to my presentation as a starting point, and have a look to the lecture on the website of the IRDIRC project he gave. And probably IDDI will invite him to give a surrogate endpoint lecture as well. So then we can learn much more about that.
Okay, it's a good idea. Thank you, Dieter.
Yeah, you're welcome.
Well, thank you very much for those answers. We've reached the end of the question and answer portion for this webinar. If your question wasn't attended to during the presentation, the team from IDDI will do their best to follow up with you after the webinar. If you have further questions, please direct them to the email addresses showing on your screen. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Professor Hilgers and Everardo Saad, by sending a quick message through the chat panel. We hope you found this conference informative. Have a great day, everyone.